Assalamualaikum and hi Madam Munira. My name is NADIRA binti Muhammad NIZAM. My metric number is 23DBT 20F 2017. Today, I would like to present a virtual lab of DNA extraction simulation. So, before we begin, we need to know what is DNA extraction. DNA extraction is a mode to purify DNA by using physical or chemical modes from a sample separating DNA from cell membranes, protein, and other cellular components. Okay, next, DNA extraction involves lysing the cells and solubilizing DNA, which is followed by chemical or enzymatic mode to remove micromolecules, lipids, RNA, and protein. After that, let's discuss about the function of DNA extraction. Extraction of DNA is often an early step in many diagnosis processes used to detect bacteria and viruses in the environment, as well as diagnosing disease and genetic disorder. So now, let's begin our virtual lab. In this experiment, you need to isolate some DNA from a human test subject. DNA extraction is typically the first step in a longer laboratory processes. Now, let's get started. There are 4 steps that we're going to follow. Firstly, collect chick cells. Secondly, burst cells open to release DNA. Third, split DNA from proteins and debris. Last but not least, isolate concentrated DNA. These are the equipment and material that we're going to use for this experiment. Drag the swab over to the inside of the test subject's mouth to collect a sample. Next, place the swab into an expander tube. End of the swab must be cut off so that we will be able to close the tube. Place the tube into the warm water bath. This salt causes protein and other cellular debris to clump together. Place the tube into the centrifuge. Inside the centrifuge, the tube spins around at high speed. Use the micropipetter to carefully remove the top liquid, which contains DNA, and place it into a clean tube. After that, add some isopropyl alcohol to the tube. Inverting the tube several times to mix the isopropyl alcohol into the DNA solution. This is because DNA is not soluble in isopropyl alcohol. Place the tube into centrifuge and click on the lid to close it and turn it on. This time, after the sample spin in the centrifuge, the DNA sinks to the bottom of the tube. And that's it for our virtual lab. Now we have an extracted DNA. Thank you, Madam Munira.